Hello, hello, I am Joe the Science Bro, and this is Science Talk. I know it's been a little bit since we've done one, but we're back. We have joining us Dr. Balin. He has a very important article to discuss with us, one that culminates in, are we talking 15, 20, how many years worth of work is being brought together with this?
Oh, in some ways, it's probably at least 3 decades.
3 decades?
Yes.
Wow. I've pinned the DOI number, which is kind of like a ISBN number, but for journal articles.
Right.
To the chat. So if you wish to read this paper, you are more than welcome to. Review articles are kind of written in more plain English. Are they not?
They are. They have aspects of scientific jargon within, but overall the background sections and discussion are more generalized to the wider audience, I would say. Yeah.
So you didn't do this just on your own, obviously.
No.
You did this with, I read a portion of the article, I apologize, it's a very in-depth article and it does require a good bit of attention.
Yes.
You have people from Edinburgh, North Carolina, Texas, Oxford, California, Massachusetts, but also you have a research consortium.
Yes.
Attached to this as well, and they're from Australia, Southeast Asia, everywhere.
Yes, Switzerland, a number of places in Europe and Australia, as you said.
How did you get all of the? Obviously, you didn't do this alone, but how did you get all of this together, really?
So I must give a lot of credit to one of the first authors. Actually, it's the second author, but she is, and the first author are co-first authors. Nikki Schultek from North Carolina. She started a group called Intracell Research Group because she was actually a patient advocate. She had suffered from chronic illnesses and was treated for infectious disease, including for the organism we've been studying, Alzheimer's disease, the chlamydia pneumoniae organism, and got better. And so she started seeking out individuals studying chronic infections and chronic disease with at first specifically for Alzheimer's disease and this particular organism that we're studying. But then it branched to other types of diseases as well. So she's involved with asthma projects, with Alzheimer projects, and looking at how chronic infections associate with chronic disease, and that this has been potentially, we think, a missing link in understanding why so many people are developing now chronic disease and what the underlying root cause would be.
So, take us through what a consensus paper is.
Okay.
Because I know that's kind of a different paper than what we've discussed in previous years and in previous sessions here.
Yes. So a consensus paper, and Nikki Schultek actually contacted a lot of these people around the globe. And some of these people I knew as well already, but others I did not. So brought this group together and developed now a consensus approach of trying to now understand what would be the best approach to take if we were to evaluate, and will be evaluating, individuals with mild cognitive impairment or early Alzheimer's disease for presence of infectious agents that they have on board in their bodies that could actually be important for disease causation. And we're talking here about how do you sample the different fluids of the body, different parts of the body, to now find infectious agents, and how to go about using the different protocols we know how to use to now do a molecular analysis of these different fluids like blood or cerebrospinal fluid or saliva, urine, etc., to actually now give you the best approach of finding molecular signatures of these organisms, and then going about proving that these are there by doing sequences and sequence analysis and bioinformatics, comparing to existing databases of what these genetic sequences are and how they compare to what we know about infectious agents already, and then to treat what you find. So potentially we have therapeutics, antibiotics, antivirals, antifungals, anti-inflammatories, etc., anti-parasitics even at times, that could be helpful in treating what you find to see if we can improve that individual's condition. And this goes to an issue that is currently many articles in the literature have been published on finding infectious agents in individuals that were diagnosed with dementias and treating them, and they got better. So this goes to the issue of reversible dementias. And there are a number of different organisms that can actually now, that we already know of, that can lead to a reversible dementia. For instance, HIV, the human immunodeficiency virus infections in neurocognitive disease. It's been associated with that. Well, if you find that and you treat for the HIV infection, these people can get better if you find it in time. There are other organisms like the Treponema pallidum, the causative agent of syphilis. Also, it can go into the nervous system. Borrelia burgdorferi for Lyme disease. You can get neuroborreliosis and treat for that, and people can get better. There's another one, Cryptococcus neoformans. It's a fungus. And people will show up in a demented state at times, but it's a fungal infection. You find that you have a fungal infection, you treat for that, and people can get better. So we already have these examples of reversible dementias. Now we're talking about looking at other organisms that have been more associated with Alzheimer's disease, that have been found either associated in brain tissues of individuals that died with Alzheimer's disease, or that have been found on board in an individual, maybe not in their brain tissues, but in their bodies, where we think it could be involvement. For instance, gut microbiota, including the oral cavity, where certain organisms in the oral cavity have been associated with Alzheimer's disease. So basically, if we find these on board, if we start to treat for those, potentially we could make a difference in this issue.
So one of the things that I think there's a couple of points in the article that I think really need to be highlighted very big. One of the biggest ones, at least for me, is the challenging of common convention that one organism, one pathogen, one virus, whatever you want to call it, causes one disorder or one disease.
Yes.
You're now saying that these many different organisms, pathogens, what have you, are now causative agents of potentially a unknown number of disorders and diseases.
Yes, and we believe that's going to be the case. The way we have analogized this is to use the definition of pneumonia as an example.
Yes.
So people that are now diagnosed with a pneumonia, you could have a bacterial pneumonia, you could have a viral pneumonia, you could have a fungal pneumonia, you could have an aspirated pneumonia, you could have many different things that can cause pneumonia. Well, we're looking at Alzheimer's disease potentially as a pneumonia. Okay, we're calling it Alzheimer's disease. Well, you could have many different agents here, we believe, also in a disease like that. Infectious agents that could actually cause the problem. You could also have brain trauma that could lead to the problem as well. So you have different ways that you can lead to an endpoint. Right. And in many cases, you may have co-infections, polymicrobial issues at the same time. You may have other chronic conditions that also you can associate with Alzheimer's disease, like atherosclerosis and diabetes, which we talked about. Chronic brain injury or traumatic brain injury over many years, many concussions, things of that nature. So we're really now starting to uncover and unfold how infectious agents may also be involved with this issue. And that goes to more currently with the COVID-2 issue, with coronavirus V2, with the COVID-19. of how that also has now led to, in many, many people, neurocognitive change that may predispose to dementia at some point in time. In fact, in the older population, it seems like it already does predispose those individuals, and I'd say older, 65 years of age and older, predispose them to now going on to develop Alzheimer's disease. Well, is it because you're now just generating in general an inflammatory response? Is the virus getting into the brain through the olfactory passages, which we believe it is happening, but also through the blood? Sometimes you find virus, sometimes you don't, which is interesting. That goes to another concept of more of a response to an infectious agent on board, leading to inflammation that can also now create damage in the nervous system. So we're now uncovering this a different idea here. The paradigm shifting is really toward causation of how are these involved. And we know that infection, by the way, is our major driver of all inflammation in our bodies. A major driver. It's probably the top of the list. And then we have other issues that can happen in the body that also lead to inflammatory responses.
I mean, again, you know, I know I've harped on this to you since last week when the article came out, that this is really a big, major change in the way that we look at, you know, pathology, especially in science. I mean, if you talk to many scientists, they'll tell you for many, many years, we've known that one causative agent can cause multiple disorders. And it's not just one causative agent causes one set.
Right.
You can have multiple disorders that pop up from a single pathogen. But in medicine, for a long time, it has only been 1-to-1. And it almost seems like not only did you plant this flag, you planted this flag that chlamydia pneumoniae causes, you know, is found in cadaver brains of Alzheimer's patients. You planted that back in what, 2000 and, no, that was 98.
1998.
1998.
Right.
So, you know, that's close to 30 years old. You know, why is it that in medicine they're not shouting it? You know, why is this not being preached as the gospel, if you will, in medical training? I know that in science it's slow, but why is it so slow for the medical community to kind of pick this up as well?
Well, it's interesting. You know, all these associations have been made with a lot of infectious agents, including the one we've been studying, as well as herpes and Lyme and Porphyromonas gingivalis. And we believe it's because there are different populations that are affected by these different agents, okay? And those different populations may or may not show disease, like an Alzheimer's disease, when they're infected with these organisms, okay? So in all cases, you're not necessarily developing the disease, but you have these organisms on board. Because we have found them in brain tissues of individuals that had the disease, we believe it's not just a coincidental finding. We believe that actually there could be involvement with initiation events and exacerbation events, etc. And we want to take the less naive approach of just saying, oh, that's unimportant, to know this has great importance for understanding causation. And part of the reason is that we've known about Alzheimer's disease for a long period of time. And based on the length of time we've known about it and still not understanding what's really causing it, you know, it's time to move back from just looking at pathology and blaming the pathology like amyloid accumulation, tau accumulation, on the causation of the disease. Those are outcomes of cellular damage, cellular change. Now, those outcomes, as we see with amyloid, the amyloid actually can be a good thing as being an anti-infective molecule, which has also been, you know, reported in the literature for over a decade now. So it can behave that way. But amyloid also can accumulate and lead to its own level of generating inflammation by brain cells, the glial cells, that are responding to it to try to get rid of it in the brain. And you can have chronic inflammation that's subsequent to an initiating event.
Yeah.
So many of us think that initiating events could very well be your infectious agents, and how your infectious agents now lead to the initial inflammation and cellular change that starts the process.
Yeah.
Now you've started a process, and now the process is sort of taking on its own life form because of the pathology that it starts to accumulate.
I mean, we've also talked about in the past that when we attack these pathologies in the normal way that we have in terms of medical dogma, which is to apply an agonist or something that binds this pathology to kind of remove it, you and I have talked about it before that one of the newer anti-amyloid drugs actually caused leakage in the brain vasculature, which caused blood on the brain.
Yes.
Which is an extremely dangerous thing.
It can be. It's one of those adverse side effects of the latest approaches. Now, the FDA appears to be at the brink now of approving Leqembi, which is lecanemab, that Asahi Pharmaceuticals has developed, and approve it for use in early Alzheimer's disease.
Yeah.
Well, many of us find that there's a bit of trepidation on our parts of evaluating this because it does have adverse side effects. Some of those are hemorrhage and edema in the brain. For a number of people. Now, when this goes into wide distribution in that population, we may be seeing more people that start to experience that. So you have a risk-benefit ratio here and basis. Now, this is the first drug really that has come out to try to treat us part of Alzheimer's disease, we'll put it this way, in the last 25 years. Because people are desperate for something, they're desperate for this. Now, this particular drug, when they did the clinical trials, showed that there was a 27% of the clinical trial participants getting the drug showed a slowing of cognitive decline. Well, slowing of cognitive decline meant, okay, you have now maybe an initial potential blockade to some extent by extracting out amyloid from the brains of these individuals, but it's not unlimited. It's one of those things that will continue to progress over time. So maybe initially there is some level of hope, but it still doesn't answer the question. What's really causing the problem? What's really driving this to be the problem to begin with?
Right.
That's where we need to have this change in paradigm and change in focus. And the big change in focus here is looking at how infectious agents can drive the process, can initiate and drive the process. So,
again, I kind of want to go back because you're challenging, as I said, you're challenging medical doctrine. Can you give a couple of reasons why there would be some pushback from the medical community, almost challenging what you're saying? Why don't they just go, oh yeah, you're right because you're the scientist doing the research?
Yes, well, it goes to, in the scientific method of you find something and then you go about trying to prove it. Okay, so we're trying to prove chronic disease with chronic infectious agents.
Right.
And this doesn't happen overnight with the process of someone getting infected and then showing disease immediately.
Right.
Many of these organisms are chronic and latent and persistent in our bodies.
Yeah.
If they get into the brain, they can also behave that way. Now, you have slow change over time, is what we believe. So what we've done is, over time, we've looked at cellular models, we've looked at animal models, and you try to prove, using those types of approaches, that you have changes when you infect these cells or animals, changes that are comparable to what you would see in a human.
Yeah.
And we've actually been able to do that in a number of cases. But yet, proof goes then to the level of, with an infectious agent, of having a therapeutic that you can use for a treatment regimen, and you want to see if the patient gets better.
Yeah.
Does the person get better? Now, if the person doesn't get better, you're going, okay, you're either too far along in the disease process, you have not hit your window of opportunity, or that the anti-infective agent that you're using in this case is the wrong one.
Yeah.
And that goes to then our consensus of, well, let's find what we have there,
Right.
so that we could potentially use the right agent in a therapeutic. It would be like saying, oh yes, if we find a fungus on board, let's use an antifungal for the therapeutic and see if the person gets better. Versus, oh, we're guessing that they might have one of these agents on board,
Right.
and then throwing the kitchen sink at it by trying the different types of anti-infectives that
Which can also
may or may
have
not
their-
work.
And they also might have their own associated symptoms, which may
Correct.
further complicate the issue, and also potentially cause patient instability. In more than one way. We're not,
Yes.
when we refer to instability, we're not just talking about mental instability. We're talking about their health at large.
Correct. There is another rationale of why the physician community has been reluctant to put any Alzheimer individual on like an antibiotic or antiviral therapy. And some of that has gone to, oh, we'll create a super infection if we do this in this population.
Yeah.
Because a lot of them are medical directors for either Alzheimer's memory care centers or nursing homes, and they don't want something to happen with the whole population,
Right.
where you have these individuals housed, basically.
Yeah.
So they don't want to do that. There are some docs that will do this for individual patients that are still living at home.
Yeah.
That would do it off-label and say, okay, let me try this to see if this would work. In some cases, there has been success, actually, limited success, but that's more of an anecdotal type of understanding versus a very well-designed clinical trial approach.
Right. Another aspect to this that I really like about this article for me kind of hits it kind of hits all the right buttons for me. To use a phrase that my father used to love, it tickles my fancy a lot, because not only are you providing consensus in the form of education to the wider community, but you're also calling for more consensus in other areas as well. You're not just calling for consensus in just this topic.
That's correct.
I've also noticed, especially in the very beginning of the article, you're calling for consensus across the board, that we as scientists need to kind of come together more often to offer these kinds of consensus.
Yes, in fact, this approach would go to trying to understand a variety of neurological and neurodegenerative conditions, from young children to older adults. There are a number of pediatric instances where there's neurological change due to either like a strep infection and or other types of infectious processes that have led to psychiatric change in children.
Yeah.
And we call it PANS or PANDAS. And we think this whole approach could be applicable to that as well, to better understanding what's happening in these younger people as well that lead to behavioral and psychiatric changes and cognitive changes in a younger population. So this is actually happening with certain types of infections in younger people. And now we need to better understand that as well. And then we could apply this to things like multiple sclerosis and to ALS and to amyotrophic lateral sclerosis. We could apply it to depression, to schizophrenia, to bipolar disease. We can apply this to many, many neurological issues in which we are finding that you do have evidence for infectious components. For example, recently it's been shown that you can actually, by changing and using probiotic therapy, you can actually help people that have different types of neurological complaints.
Interesting.
Or diagnoses, including depression. And part of that is probably of what's happening in the gut with microorganisms, because they can produce hormones, neurotransmitters, different types of molecules that are useful for us to keep our cells healthy. And so by knowing this, we need to focus here we now have these trillions of organisms in and on us, and that we're exposed to through our environment. We need to understand this relationship. Right. Because the relationship there is actually impacting our health and sickness.
So, one of the things that kind of pops into my mind whenever we discuss, like diet, is how important, the research that's going on in the gut microbiota, which 10 years ago no one was talking about.
Right.
At all. It was that was before we constantly saw Jamie Lee Curtis, on the Activia commercials. That was before we saw all these commercial products for just little probiotic and prebiotic pills that are available. How important is it for different topics of science to really look into how other areas affect the wider issue that we're looking at? Because take you and I, for example. We work both in brain, but I also work in the bone environment. How important is it for, like, let's say that relationship, the bone environment to the brain environment?
Yeah, so that's really important, because what we're talking about here is systemic issues. It's not just organ site issues, but it's really the whole body approach. This is why being in an osteopathic medical school is ideal, because it's a holistic approach. It literally is a holistic scientific approach here of looking at a person, looking at their body, what's happening. Much of this is systemic, and we need to look at it that way. What this is doing also is taking us out of a silo and putting the silos together. Okay, that's consensus. Okay, let's use infectious disease people. Let's use neuro people. Let's use inflammatory people. Get these people together and saying, okay, what are we studying? How are we studying it?
Right.
What will it lead to if we do this in a collaborative sense? That's why it's so important in crossing over in all these disciplines to understanding how to actually do this, how to collaborate, and to call for that, and to get people together to do it, so that if you have an expertise in, for instance, in infectious disease, and I have an expertise in neuropathology, okay, let's use the expertise, let's bridge it. Let's say, okay, you know how to do things with infectious disease, I know how to do things with the brain. Let's see, how does that combine?
Well, there's also a lot of overlap in a lot of these topics. I mean, we talk about the brain and the bone, just for that example. There's vascularization, and there's vascular differences there that are very important to each other, because, I've always told students whenever I've taught them, the human body lives on a very fine line of balance, and if you mess up one organ system or one part of it, it throws the whole thing out of whack.
It's so true.
Yes.
It is. I always tell their students too, it's all about homeostasis.
Yes.
The balance, as you said. If you don't have a homeostatic nature to yourself, something's going to be different. Something's going to go wrong, probably.
In a way, that's kind of what medicine is trying to bring you back to that balance, that homeostasis.
It is. and for the most part, most of us feel like there's a homeostatic balance. It's sort of like, okay, so as an example, I guess, it's like, okay, one night you only get 4 hours of sleep, but the next night you say, oh God, I got to get 8 hours of sleep, because I've got to maintain the balance, right? And sometimes, it's like catching up, and you feel like, well, I can never catch up. However, you do have to try to maintain a level of balance. Fortunately for us, we have adaptability as well. So we can adapt to some changes in our environment, adapt to changes in our diet, adapt to changes in circadian rhythms, things of that nature. But even though we can adapt, there are going to be things that we can, keep us in balance or not. If you go out of balance and you stay out of balance, it's probably not going to be good long term.
Right.
It could lead to something that's more chronic then, and something that may emanate from this chronicity into a disease entity that gets recognized later on.
Right.
So,
one of the aspects to consensus that I think is rather odd is when we're teaching new scientists, especially when we're in the classroom, we always preach to them consensus is extremely important. If you don't have consensus or unification of purpose or theory or hypothesis, you can't move science forward. But in practice, we rarely, as a greater scientific community, really do come together in a way that you have in terms of consensus. There's really very little unification of theory almost sometimes, which, again, I have to applaud you for this article for that. I'm not just, kind of trying to blow smoke up, anywhere, because it's truthful. This is something that I really haven't seen. I think I might have seen one other consensus article that even came close to the widespread that you kind of have in terms of researchers.
Well, it's true. What's interesting is that there is now, and there has been for a few years now, more calls for consensus in a lot of approaches in different arenas of medicine, health and science and medicine. And it requires an active process.
Yeah.
You actually have to reach out to your, at times, competing scientists, the competitors of research, right?
Yeah.
And talk to them about collaborations. You need to open that door. You need to realize that there's not a single individual or single laboratory that can answer all these problems.
Right.
And address them all the right way. So by bridging laboratories, and it comes down to this, is that for the longest time, a lot of people felt that, well, they wanted to have this credit given to them.
Yeah.
And more of this assignment of, oh, you're the first, or you're the one, and you're the key element here.
Yeah.
And many of us have felt that that's the wrong approach to take.
Right.
We've been stuck in where we are in doing the science we do, and we've collaborated with other people.
Right.
But not to the extent we needed to. So many of us have been calling for this and saying, okay, let's reach out. But we also need people that can organize that.
Right.
And that's where Nikki Schultek comes in, because she's a great a communicator. She is a great person to reach out to others and say, oh, do you know this person? If you don't, this is an interesting person to know that can help us with.
What was it? A consortium.
Right. It's the Intercell Research Group Consortium.
How? Number one, how does a research consortium even work? Whenever you hear consortium, a lot of times it's in the connotation of some villain in some story somewhere.
Well, in this case, it is really an agreement. Now, it's almost like an informal agreement. There's nothing written here that we have to agree to, in essence. It is an informal agreement of individuals that have a like-mindedness to achieve something. And what are we trying to achieve? We're trying to achieve in a way, a process that will lead us into a path of greater understanding and acceptance that we're now bringing a whole different shift in our world of understanding to this major disease, which may be applicable to so many major diseases. And we think that this is a requirement at this point in time, that too much time has passed where this has not happened. So that's why we have the people that have agreed to be part of the consortium and agree and worked on this consensus manuscript for months to really come up with this approach to actually get it into a published arena here where people can take a look at it and say, oh yes, I agree with this or I don't agree with this. And if you agree with this, would you like to be part of the consortium and approach these issues this way as a huge group?
Yeah.
Because that's the only way things will get done here, is to do this as a group, a large group, and and then seek out funding for these projects. There's a lot of private people that feel that they want to actually fund this because they believe that yes, this is the path to follow. That the current paths and the old paths that have been followed have not led us to answers that are really helpful for us.
One thing that I have felt, and correct me if I'm wrong, or feel free to comment on this theory. I think the past almost 20 years, we have, as a scientific community, almost become more isolated from each other over the past several years. Mostly because we're all running to be the first. And instead of trying to come across as a team or as a group, it seems more like we're doing the 30-yard dash. And whoever gets across that line first gets all the credit for all of the work, even though all of us are contributing. It seems as though I've noticed that I'm starting to see a change. That a lot of the articles that are coming out, I'm seeing less one single country being on there, and I'm starting to see broader strokes of these research groups. Do you feel similarly, or do you think that's different, or maybe it's just that it's not being shown as often? It's not getting the attention that is necessary.
Well, I think that we are broadening now more of an international community, because the issues we're talking about here are international issues.
Yeah,
and not just isolated to one country, or that's their problem, not anybody else's problem, that kind of thing. And I think that globality is now really a driving force for how we need to make this change. Okay. And I think the last few years, COVID had an impact on us of keeping people into isolation, basically. And coming out of that, I think it's also another realization that it has led to, and that is that, okay, we can't do this in isolation. We need to be interactive. We need to do this in an international sense, in a global sense. This has to be driven by really intelligent people that are in many different parts of the world. People that have gone into science and medicine and research, literally are some of the brightest minds for discovery, for experimentation, for creativity. Okay, it's not that other fields don't as well, but these are fields that have attracted that and brought people together that way. Well, let's use that, but how do we best use it? So instead of I'm going to stay in my own little room over here in the segment of Philadelphia and not interact with anybody else, that should never be the case. Sometimes, we get so fixated on what we are doing in our own laboratory, we don't look around us and say, oh yes, but what about over here and there? But what I'm finding is more and more people contact me. I'm contacting others as well. Others are contacting people for us and coming back to us. So we have now more of a group that's working together this way, which very interactive.
Yeah, I've noticed that in a way there's kind of a different shift occurring. When I was a younger student, you would have labs that were across the hallway that were basically studying almost the same exact thing in different ways, and they'd never talk to each other.
Right.
And in fact, they'd almost, I don't want to say they'd actively sabotage each other, but they'd almost actively pick each other's theorems apart, almost for the fun of it. And now, one of the things that I'm noticing is that we are crossing the hallways more often. We're talking with each other. And I think you're right. I think the isolation that we had during COVID kind of made us all remember, interaction is kind of a good thing.
Yes, I think so as well. Also, most of us who have come together in this consortium now we can be critical of each other in the work that we do, but there's an acceptance of that as well. Because the view has changed into one of I'm right and you're wrong to, okay, folks, there's a rationale behind why everyone has been studying something and have found evidence for whatever. And now, instead of saying that that's wrong, you've done something wrong to get that, there's an acceptance that that has occurred.
Yeah.
So that once that has occurred, now there's an openness to, okay, well, let's understand it. Why did someone find this organism versus a different organism when they analyzed brain tissues?
Right.
And then somebody's handing off the same brain tissue to somebody else, and they're finding something else as well, and you're going, well, wait a minute. How is it that 3 different labs found 3 different things? And then you look at, well, how did they do the science? How did they evaluate what was there in the tissue? What experiments did they do? What protocols did they run? That goes to the consensus, okay?
Yep.
Is there a consensus we could use so that all 3 labs then could find all 3 different things, right? And that's what we're aiming for. And so there's a greater acceptance up front that this is occurring. And now let's get to the root of it. Let's really find what's there. And if there's multiple things there, let's entertain what are the multiple things that are on board, and could they be causing the problem? No.
So another part of this that I really think is very important, and I feel necessary to almost read it verbatim from your article here.
Okay.
And that's Future Direction, a multi-center study that tests post-mortem biosamples using different methods will help develop consensus diagnostics to reveal most microbes. This can be used prospectively in patients with mild cognitive impairment or Alzheimer's disease to reveal potential microbial infections. And, there's more that goes on to this.
Yeah.
You're almost challenging the rest of the scientific world to join you in consensus. You're calling for more work to be done. You're not, and that's one of the things that I think is very different from your normal review article, because your normal review article is almost an endpoint.
Right.
A lot of times, we like to think that. I like to tell students that a well-designed study will almost ask way more questions than it began with. And most review articles are almost a terminal point that they don't raise many questions. They're reporting on what's already been done rather than inspiring more work.
That's very true. In fact, in this article, as you've seen, we lay out a basically the way this should be done. So we lay out the paradigm of what you should start with, or what we think you should start with, and then how you should start doing the different experimentations and different analysis, leading even to clinical trials or treatments at the same time, to really start to make a difference in the population.
Right.
Because we've realized that proving that a chronic infection is causing the chronic disease may take any one of us even much longer periods of time to actually now conclusively, without any doubt whatsoever, that this infection causes this disease. And it may take another 50 years, and then at that point in time, you've already lost millions of people to the disease. So now we're going, okay, instead of doing that, let's do something now. What are we doing now? So we're saying, let's use a scientific method, but at the same time, when we find something, compel people to start treating for it, especially if it's showing up in persons with cognitive deficits. Do something about it so that we maybe can stop the process. or modulate the process so they don't go on to full-blown disease. And this is their endpoint, is becoming vegetative
Right.
with Alzheimer's disease. Now we can start applying this to the other diseases, which we've mentioned as well, so that maybe we can start making a difference too with those other diseases, so that, or other conditions that can improve the quality of life for all these people. I mean,
Right.
that's what it's all about. We're trying to do something for humanity, improve the quality of life for tens of hundreds of millions of people, really,
Right.
that suffer with all of these different types of conditions.
And I like it in a way because a more conservative scientist would probably read some of this consensus and call for collaboration and say your hubris is showing because you're basically throwing down the gauntlet to other scientists in the field and saying, look at our opinion and try and prove us wrong. And
Correct.
Like I said, some conservative scientists would look at that and almost go, "Are you getting a little big for your britches there?" And I found myself as I started, because I jumped around the article like most people do when they first read an article, and I found myself going, "No, I kind of want to. I either want to. At times I felt I wanted to prove some of it right, and at other times I was like, no, I want to see if this is correct."
Yeah. Which is what's interesting here is that what we're actually talking about is something that really hasn't been the focus for this disease ever.
Yeah.
It's always been on the periphery of, these interesting findings over here on the periphery of, look at what these scientists have found, but we're still going to focus on our pathology and go with amyloid and tau pathologies as if that's the most important thing.
Right.
And everything else is secondary or tertiary to that.
Right.
And we're saying, and we are throwing down the gauntlet in many ways and saying, okay, folks, it's time that we start taking a realistic approach here. What's the reality? The reality isn't that, all this work done on pathology, which has been great. That is an outcome. This is happening. We're saying, okay, we're going to study why it's happening.
Yeah.
What is the rationale behind it? Okay. What is the factor, or what are the factors that are really driving this, causing it and driving it? And that's what we really need to understand. And as more and more work has been done over the years, we've recognized that the immune response and the inflammatory response is so prominent in this disease and in others. And so it's like there's a lot of information that's been associated with getting to pathology. But we really haven't understood why that's there, why it's really now involving your immune system, your inflammatory responses, your cellular change up front, why specific regions of the brain are being affected early on in the disease. What makes sense here? What is the real rationale behind?
Absolutely.
That's what we're doing.
Absolutely. And another aspect to this that I have to compliment both yourself, Nikki Schultek, I believe Dr. Lath, I believe that's how you say that. I have to commend you.
And others.
And others. I have to commend you on the way that this is written, because it's also written as you have made yourselves experts on this topic in its current form. And you are quite literally laying a foundation for what comes next with this article. And again, I have to commend you on the way that it's written, because even for a scientist, myself, I found the writing of the article, and again, I'm not trying to simply, shoot a praise out into the air, but I found this article easier to understand than most review articles that are out there. It is relatively free of a lot of the jargon, which you and I have talked about before, and it doesn't put people in a position where they're not going to understand it if they're not plugged into that particular community.
Yes. In fact, many of us worked on this article Obviously, I am one of many contributors
Yes.
here as well on this article, and even in the consortium that's listed at the back of the article, the names of those individuals as well. Also reviewed this article, gave criticism on the article, edited the article, etc. So this was actually something that there are. I don't even know exactly how many people are on this.
A lot of your collaborators are listed here, and there's a ton.
Oh yeah, and so everyone that's on this article looked at this article, reviewed this article, had input into the article, and then it came together. And Richard Lath, Nikki Schultek are first authors on this, because they were taking this information and starting to mold it together. And they're both excellent writers. They're both excellent in putting information together. Okay, so they're taking all of this, and it was sent back out a number of times for us to re-review.
Yes, I remember you. I
and
think I remember you talking about this in
Yes.
July of last year. Actually,
Well, we put a lot of effort into this,
Yeah.
and it went to a point where, by the time we've all finished this, when it became a finished product, it was reviewed by so many different scientists with different writing styles, but that's where the first authors put it together to make it a cohesive article here in a consensus paper and review paper, obviously. That now, I think that when you pick it up and you see what's in there, you realize that we're talking about and using evidence already that's in the literature, and talking about this in a way that I think makes really good sense to anyone picking it up of what our approach is and what we're trying to accomplish.
Right.
And it should be a one of those articles, I think, that will be now referenced as this is what needs to be done. This has to be done. This needs to be followed up on. We will continue to do this work, and others, I think, will join us as well along the way.
Yeah.
And by doing that, we will come to an understanding here of how to approach infection, this disease, and other diseases like it.
Absolutely, and another aspect to this that I think also needs to be kind of addressed is that you are actively saying that it's not just the neuro people that need to be looking at this.
Right.
Correct. It's all types of scientists. It's people that are working on the blood-brain barrier. It's people that are working on traumatic brain injury via cranial dysfunction. You're calling on basically everybody in the scientific world to look into this and say, there's something here.
That's true. We looked at that too and considered that we need molecular biologists. We need infectious disease specialists. We need all kinds of different brain specialists, neurologists, neuropathologists, neuroscience in general. We need bioinformaticians as well. Okay, like you. it comes down to that we need this because it will help us get to the best answers. Okay, it's solving the problem using all of this brain power in all these different disciplines to really get to the root of what's really happening. And that's why I'm so excited about it, because I really believe this is now turning the page on this type of issue to opening it up to and say, okay, we need you to look at it, evaluate it. If you want to be critical of it, be critical of it in a negative way or a positive way. Okay, but you know what? This is what we're throwing out there. This is what we believe truly needs to be done to get to the answers, the final answers that will make a difference for the population.
And that's another thing that I also enjoyed a little bit in the very beginning. You're highlighting everybody's work in terms of what the current state of the literature is, but you're also not shying away from saying, hey, we've had some failures in terms of what we haven't looked at yet.
Oh yeah.
And that's something that you don't normally find in a lot of these articles is, we don't point a light at the shadow in the corner because, we're trying to tell a story about what it is that we did, but you're actively going, no, we need to see what we haven't done yet.
Oh, absolutely. In any research effort, well, in anything we do, actually, we don't necessarily have all the answers up front. We have approaches to be taken based on what we've observed.
Yeah.
And we're trying to now get the best inputs based on that. And the input of how to proceed, of what to proceed. If there are things that we haven't mentioned there, and somebody else says, "Oh, you need to also consider this."
Yes.
Yes, absolutely. Open that door. Let's consider that. Let's get into that. Let's bring that into part of the equation, because we don't have a final equation here.
No, you're right.
We're developing this as we go along. At the same time we're doing our work, we're developing, okay, the paradigm is shifting, but the paradigm is not complete. It is still it's evolving and developing. And that's the way we're looking at it.
No, you're right. And we touched on this, I think, in our very first talk together, and that was the black box, the gray box, and the white box theorem. We know there's something there. It's the gray box. We know there's something there, but we just don't know how many steps are involved. We don't know,
correct,
if there's multiple causative agents. I mean, you and I have talked ad nauseum about the idea that chlamydia pneumoniae is just sitting there causing these repeated insults that don't really cause major issue, but then something else could come in and disrupt that balance, and now suddenly everything is, the apple cart is on the side of the road.
Correct, and it's the right. I think it's the correct way to look at the problem. We can't just start assigning that this is where it is and that's it. All of this is part of the this evolving paradigm, and we will be able, I think, eventually to understand how things sort of fit together. When we get down the road on this and start doing evaluations that are even much more intense and complete and thorough, using even other methodologies that are still, those are even evolving as well. So in doing this, we have this excitement by so many people to actually do it, to really now start applying it in this way. That's what's going to make the difference. It truly will, because of all of this input.
I do think also there's a benefit here because of the consortium, the research group. It makes it so that if there is someone who has, let's say, that is privately wealthy, and they want to donate money for this, it makes it easier, because that almost is the big blinking light in the sky going, okay, there's the point that I can donate to, and that money can be disseminated to people that are doing this. I was having this discussion with another researcher earlier today. It's very difficult these days to get research funds. It doesn't matter what the topic is.
It is. Well, part of it is that there's so much research being done.
Right.
And there isn't, if you have, our governments can't fund all the research that's being done. That has become evident over the years. So that doesn't happen. So we require to have private funding, individuals that have an interest in a particular topic to actually, that have a lot of money, that can afford to fund aspects of these projects. In ways it is a requirement, and by the way, there have been people already coming into this that have been doing it, that have already now looked at what is being done and said, yes, that has value. That has value to make a difference. I want to put money behind that to see the science being done, which is a great thing. And that's the way this is going to be done too. And so those individuals also need to be given credit for actually recognizing what's important here, what's going to make a difference, and saying that's where I want to help. We're all in this, so we're all using what we know how to use. So many of us who have gone into science, we have for whatever reason an ability to do this type of work.
Right.
So in some ways, we're using that God-given ability to now apply it to this.
Mm-hmm.
And there are a lot of us that do that, and by other stretches of effort from others that have made money in a different field, but recognize that healthcare, these particular diseases are so important to understand, and they want to give back that way, which is super.
Yeah.
So we have to give a lot of credit to those who are funding these types of efforts.
Mm-hmm. One thing that I've noticed, especially when you look through certain aspects of history, it seems that those who scream the loudest for innovation in these research fields are the ones who have already benefited from what has already happened. And they know now of the deficiency that occurs. We talked once about I forget what the name of the researcher was, but he was doing Alzheimer's research, and then he gained it. It started developing the pathology, and what he did was over time he started painting his portrait, and I think he did it once a year. He would keep painting the portrait. And each year, you would notice detail would be lost, and then it would start to become more abstract. And mind you, I should mention to our audience, he wasn't doing it with a mirror there. He was doing it off of memory of himself.
Yes.
And I believe at the very end, it was just a conglomeration of circles.
Basically, that's true. And it comes down to it demonstrated that in being able to do a self-portrait, that he lost the ability to even understand what he himself was as a human, and how he could even depict himself that way. It became unrecognizable.
I almost liken it to how dirt and fog and dust and moisture can build up on a camera lens. It becomes more and more blurry, that you lose more and more detail as it goes on, and then suddenly it's like, I think that is something, but I can't tell for sure.
Yeah, and it demonstrates in a way, in a very dramatic way, the extent of brain damage that occurs in the disease.
Yeah.
And it is dramatic. It's throughout. At the end, it's so many different areas of the brain are affected. It just is devastating.
It's one of those disorders
process.
That at the very end, that's where the sheer magnitude of what has occurred really shows itself. With a lot of disorders, the symptoms are all there throughout the entire thing. It's very rare that you develop heavier magnitudes of the symptoms. It seems this disorder, this disease, because it's degenerative in nature, it really gets worse as time goes on, to such a degree that at a certain point, what was the movie? Benjamin Button. At the very end of the film, he's a child and quite literally has dementia, which reverts him to a very childlike state.
Yeah. In fact, many people have described it that way too. That it's like having now an adult, but that is now at a childlike behavior, and can go even to almost an infancy type of behavior, where it's total dependency, unfortunately.
Yeah.
But it can be a process that takes 20 years to occur in some people. And that's what's agonizing about it too, because if we have no way to really efficiently treat that and stop it, it's basically a process that you see the withering away of the person in essence.
Yeah.
Which is it's one of the reasons why I came into doing Alzheimer's disease research to begin with, is that I had an uncle who actually started experiencing it that way, and actually at the end was vegetative.
Yeah.
Ended up dying that way, where he had no cognitive ability. And I thought, well, this is the cruelest type of disease that you could ever imagine.
Yeah.
At those golden years of age, to have that happen and that be the outcome, that is unacceptable.
No, I agree with you, and it's in a way it's almost, it cheapens all the work and all of the things that came before in that life. I don't think that a lot of people really understand just how devastating something is like this until you actually experience it for yourself, because quite literally, that person that you've had throughout your entire life, at one moment, it's just gone.
Yeah, and for the person to be gone is other diseases. Well, there's some other diseases too that can lead to that, but this is one of the most dramatic ones that does. In other types of diseases, unfortunately, a person still has a mental capacity, cognitive capacity to interact, to still be a person. And yet, they may go into demise as well, but they're still a person.
Yeah.
In this case, you're losing the person. You now just have a body that is potentially still functioning, but where is the person? It's no longer there, really.
Another aspect that, and again, I haven't read all of the papers, so I really should say that to everybody. The way that I tend to read papers is I bounce around as the titles grab me. One of the things that I noticed in here is that you actually describe the symptomology as it's not the same for every single patient.
That's
which
correct.
Is another convention that is kind of being challenged more and more these days. I've noticed in journal articles, in case studies of patients, and just in anecdotal reporting that I've noticed as well, is that we're starting to say you're not always going to have a fever when you're sick. You're not always going to have a cough when you have a cold. You're not always going to develop phlegm when you have the flu. We're now saying these are collections of symptoms. They're not all the symptoms that you're going to get.
That's true. And each person will sort of evolve in a way with the disease. There could be a lot of similarities, but there can be significant differences as well. And some of that is because of how the brain is wired.
Yes.
The nerve connections, which nerve cells now are damaged, and which pathway is being used in the brain. So some pathways, there's some commonalities, and some of those are short-term memory issues start to show up, spatial issues start to show up, where people getting lost and things of that nature. There could be specific cognitive deficits that show up, like unable to do math, or unable to now read a paragraph or read a book or whatever. You could have a different level of aphasia, things of that.
Yeah.
So there are different symptoms that can show up depending on which area of the brain is being most affected and then how it progresses. And that can vary. And part of that is the connectivity of what is being really damaged and then what does that control, obviously.
Well, the brain, as I understand it, has a lot of backups in it. It's not just a single pathway as a single thing.
Right.
It's very much like you have multiple routes to the same aspect. So it does take a long time to see these noticeable differences. And I've seen a couple that say that it's actually a slowing of the rate of cognition at the very beginning. And then it's an inability to have the cognition. And then it can be memory, and then it can be motor function. Even now, I believe that there are cases being reported in Parkinson's where they don't even have the Parkinson's shake anymore.
Right, you can have a variation on a theme. That's true. Like we call it maybe a Parkinsonism.
Yes.
That it's giving you symptoms of Parkinson's. Is it really Parkinson's disease or something else?
Yeah.
And it could be something else. And at times that's why it's difficult. While we assign diseases in life based a lot on symptomatology. And some diagnostic capabilities, either brain imaging or analysis of fluids and biomarkers, things of that nature. There's still a number of diseases that cross over with symptomatology that we may not know until death. And then you do a post-mortem evaluation of brain tissues, and you can see in different regions what type of pathology was there. You can associate it back to the symptomatology and then get a better understanding of what the designation of that disease really was. And basically it's just sort of like highlighting it that way based on pathology and symptomatology. But you're right. There's a level of crossover. There's a level of also progression differences, depending on the type of problem that's occurring. That can also occur. We try to best evaluate. A clinician will best evaluate the individual based on testing, often neuropsychiatric testing, things of that nature.
Yeah.
Of what they think is the likely diagnosis. And often it's correct based on what has been seen by others as well,
right.
And making that diagnosis, and then through the autopsy and post-mortem examination of brain tissues, revealing that yes, that was the case.
Yeah. One aspect that I wanted to discuss with you is, have you noticed, at least in recent times, that there's kind of been a move away from doing this kind of research in an academic setting and moving towards privatization of doing this work? Or have you noticed it actually shifting in the opposite direction, that it's moving more towards academic research rather than private?
That's a good question. I actually, for this research, there's a lot that's gone on in the pharmaceutical companies. I know that. As far as biotechs, a number of biotechs have approached the problem too. A lot of it's been geared toward diagnostics.
Yeah.
There are a lot of clinical trials, though, that are undergoing, that have been started for Alzheimer's disease and other neurodegenerative issues, looking at other pathways and what pathways might be involved. And even some clinical trials on anti-infectives have even started. Some of the antiviral clinical trials based on the herpes issue.
Yeah.
And herpes simplex being involved as well. And even on some of other organisms. But we need much more of an effort in those types of clinical trials. Right. As far as privatization goes, biotech, big pharma, there's still a lot of work being done in academia. Okay, so many of us are academics still.
Yeah.
So that is occurring. The NIH, also through the NIH here in the States, there's work going on at the NIH and the different institutes. So I don't know if I've really sensed that there's been a shifting of that. There's been at times a focus of academics working more with biotechs, working more with diagnostic companies, trying to now work more together in a collaborative fashion. But in some ways, that's why this consensus paper actually is calling for more of that even.
Yeah.
So it'd be a public, private, academic, pharma kind of approach, and NIH type of an approach to all of this effort.
Now, you had a recent experience where you were a part of a panel.
Yes.
On this topic. And I believe it was sponsored by Duke University.
Yes, that's correct.
Could you enlighten us on some of that?
Yeah, so the Duke University, it was a symposium, mini symposium actually, about a half a day symposium that was sponsored by the Benter Foundation and Leslie Noren's GermQuest, which is another foundation. So Bill Benter and Leslie Noren's really sponsored it, run through Duke, and it was really had different speakers talking about different issues with infectious disease and Alzheimer's disease. That was the focus. So I sat on the panel discussion at the end of this, where we discussed why this was so important, what is being done, how we can go about actually looking at this as more of a collaboration as well, calling for collaboration, and also talking about aspects of what is being done currently and what can be done. And a lot of interesting things came up out of that panel discussion of finding that other laboratories are looking at genetic components that would apply to infectious disease, for instance, looking at microbiome analysis, looking at the better techniques to use, like next-gen sequencing or metagenomic sequencing of genetic material that is extracted from the Alzheimer individual to find infectious agent, for instance. So a lot of that was opening up that conversation and talking about it. So it was one of those things where also the Alzheimer Pathobiome Initiative that Nikki Schultex has been instrumental in starting was also involved with. That's why many of us were panel discussants about this, because we've been working in the field for a long period of time and are asking questions. So I asked the question about whether anybody really had looked at infectious agents with familial Alzheimer's disease, which is the genetic form of Alzheimer's disease, which occurs in about 2.5% of the entire Alzheimer's diagnosed population. So it's a smaller segment, but it's where you have genetic mutations that lead to Alzheimer's disease 100% of the time. But could there be infectious components there as well?
Yeah.
And basically, it went fairly silent when I asked the question, because I don't think anybody's really looked at that issue in those types of brain tissues and that analysis of those individuals. And I think that should be done as well, because it's not as if they're living in a sterile environment and they just have a genetic disease here. No, they have other components as well that could be actually now contributing to this outcome as well. So,
no, that makes a lot of sense. So, if you could, what's just for the benefit of our viewers, what's the difference between a symposium and a scientific conference?
Well, a symposium usually is more of a focused event where you have a number of speakers. It could be an all-day symposium, it could be a half-day symposium where you're now speaking on topics that are all relative to one another and relevant to one another as well. A scientific conference really is more of a wider, in-breadth type of an approach, where you might get all types of different research approaches being done in a particular field. If it's an Alzheimer's conference, you may find there would be talks on infection, genetics, environment, people may be working in cell cultures, in animal models, in humans, etc. It may go into multiple different pathways that are being evaluated. So it's much more of a broader type of approach.
So it seems like symposia are very much like the local club meeting.
Yes.
Of a wider chapter. If
Yeah,
you will.
you could look at it that way. Yep.
How important are these symposia? And is there a need really for, let's say, like the general public to be involved in some of these?
Well, many of the symposia, so you could have involvement with general public to listen in for the most part, but a lot of these are really scientific talks about the research that's being done. So you're using a lot of scientific jargon and a lot of experimental jargon that many of us would be aware of and understand, whereas the lay public may not understand those types of issues. The overall concept, though, would be understood. A lot of this is based on a background of something, and now they're applying now experimental paradigms to studying that particular issue.
Right.
So in some cases, that would be the way it's approached. Other cases, other symposium often will give you a general overview of more of what has been done in the field. In fact, in 2014, I actually organized and ran a symposium through the Franklin Institute here in Philadelphia on infection and Alzheimer's disease. And I actually contacted a number of the people doing work in the field. They came into Philly, and we had a symposium, and I led the symposium, and then for a full day, I had different people speaking on what they were doing. And it was actually open to scientists as well as the public.
Wow.
There were a few hundred people there, and they got an overview of, this is what we're doing, this is what we're studying, this is why we think it's important. So,
did you get much in the way of interaction from the public?
Oh yeah, there's tremendous interest. In fact, typically the comments I get back from individuals that hear about what we're doing and when I speak on it, they're going, wow, I never knew that this could be how this disease really manifests.
Yeah.
Especially when I talk about how things can involve olfaction and come into the brain
Right.
through our sense of smell, and bypass the blood-brain barrier and get right into the brain tissue proper from olfactory bulbs and olfactory tracts. And boom, once you're there, you're right in the areas where Alzheimer's disease starts.
Yeah.
And some other diseases can start there as well, because of the connectivity, the structure-function relationships, which I've talked about often.
Mm-hmm.
Well, a lot of people are not aware of that.
Yeah.
And when they hear it, they're going, "Oh my God, that's intriguing."
Right.
It's also exciting to know that you're following the pathways this way, and that this could be an answer that we're looking for. Now, and then hearing about infectious processes in the disease. Now, it's become more relevant because of COVID.
Right.
Okay. Prior to COVID, people would think, "Oh, that's really interesting." They didn't really know how it fit. But after
Right.
COVID, they're going, "Oh my God, this is become relevant."
It does seem as though the general population has kind of become a lot more literate in diseases and pathogens and viruses as a whole. I know recently on HBO there's The Last of Us, which was a video game that
Yeah.
described a fungus, taking over the human body and causing reanimation. Well, in a way, reanimation. There's been, obviously, since George Romero did his Night of the Living Dead, we've had zombies for decades. This is one of those things that kind of, in a way, has fascinated people, is these diseases of the mind that cause us to change or alter our perspective. It almost lends the questions to, are there psychological factors or environmental factors, if you will, that go into these disorders and symptomologies almost appearing out of nowhere, if you will. The physical change is predicated on something that is non-physical.
It's very true. So when you're talking about that, Joe, what I think about is the Human Genome Project.
Mm-hmm.
And why do I think about that? It's because when the Human Genome Project started and then was finished, and you had the human genome sequenced,
Yep.
everyone thought that understanding the human genome would be the be-all and end-all to understanding disease.
Talk about hubris.
And correct. And I've always felt, okay, you've got now nature and nurture.
Mm-hmm.
All right? Okay, what is nature? What is nature? It comes down to our environment has never been approached the way we're approaching it in this day and age as ever before.
Yeah.
Why is that? It's because our environment is actually having a tremendous impact on whatever genetics we have.
True.
Good genetics, bad genetics. It comes down to the environment is such a force about us, because the environment is everything.
Yeah.
From our drinking water to our food to the air we're breathing, how the interaction, our structures around us, how we walk down a flight of stairs and stay upright. I mean, it comes down to our environment is key to our functionality.
Mm-hmm.
And both at a genetic level, but also at a gross macroscopic level as well.
Yeah.
And that is what we need to appreciate more. So all we're saying here really is we're going to appreciate our environment by the infectious world environment around us. And that's only part of it.
Yeah.
We've got pollution. That is, also a major driving force. As climate change is occurring around us, we have temperature changes that are now influencing us as well.
Yeah.
Our water is being contaminated with plastic. That's going to also influence our diets, our food sources, etc. All of these things have an impact.
No, you're absolutely right. I when I was an undergraduate, a lot of my professors were showing us documentaries after documentaries about different things. There's a documentary that I got to watch when I was an undergraduate that was called King Corn. I don't know if you've ever seen it. I recommend that you see it if you haven't. And one of the things that they did was that they clipped their hair, and they actually had their hair analyzed, because in your hair is kind of like a fingerprint of what you've been doing, eating, how your body has been. You get so much information from hair. And one of the things that they said is that you are, I think it was somewhere in the range of like almost 60% corn, because you've consumed so much corn and corn products over your lifespan that you've actually picked up certain aspects from this. So everything that can be traced back to corn.
I could be a bit corny here and say that's amazing. But I won't. That's a good point.
You know, it's 5:43 in the afternoon and he's still throwing zingers out like it's...
Well, we have to have some level of humor as well.
No, you're right. But it's very interesting what you're saying, that our environment, as we've changed it, because human beings are the only organism on the planet that modifies that adapts the environment to them, not the other way around. Although that can't be said anymore because of new research that's been done in amoeboids and different bacteria and parasites, because they are actually actively modifying the environment that they're in. And again, that's a paradigm shift. When I was a student, amoeboids were considered separate organisms that could potentially be parasitic. And now even parasites, that definition is being recategorized because we're adapting our way of thinking. You know, we're thinking about it differently.
Yep, it's true.
Mm-hmm.
We are.
I mean, you and I touched on this in the very beginning. Research has changed a lot from when I was an undergraduate back in the late double aughts to now. In 2023, research has kind of changed so much. Before the live stream, you and I were talking about how I feel the modern lab, every single modern lab is going to have a 3D printer for them to prototype and make specific things that they need specific to their needs. It's gotten so cheap. And part of that is because of the ubiquity of the thing that is being used.
That's a good point.
You know, it's not a patented thing that's been held really tightly. You know, everyone's kind of, been able to use it. I remember when 3D printers were the size of oh God, and this is really going to show how old I am, a Mac machine. I say that to some of our students, and they what?
Right.
The ATM.
Yeah, correct.
I remember that they were the size of a Mac machine, and they could only make something maybe about this big. And now you can get multi-part items that took less time than that original gigantic thing even took to make one part.
Sure. Good point.
You know, we evolve in our technology, and again, I think that's another reason why this consensus is so important, is because it might inspire that younger scientist who has the idea of the newer technology to then take this in some other direction and analyze something in a new way that we never even thought of.
Oh, absolutely. And over time, that's what many of us have done. We've added to the scientific approach by adapting the newer technologies to what we do.
Well, when you originally discovered the pathogen, you weren't even looking for chlamydia originally.
No, not really. It was on a whim, actually, to look at and ask the question about this. So it was really based on just, talking with my colleague, Alan Hudson, and coming up with, let's just try it. Yeah. I've got brain tissues. You're doing PCRing. Okay, let's try it. And he was PCRing for a number of different organisms, and he happened to have primers for this particular chlamydia organism, the chlamydia pneumoniae.
Yeah.
And we said, okay, and that's what the hit was. And then we still needed to go through this process multiple times because it was phenomenal that you would have a hit based on basically just a guess at what could be present. Right, and then to say, okay, well, do it again, and it shows up again.
Serendipity.
Serendipity, and it's interesting because science is still done that way.
Yeah.
through a lot of serendipity.
Yeah.
You have to have it's sort of like you're recognizing at a point in time, you're
Mm-hmm.
asking a question, you make an observation, and then you say, well, I think that might be important.
Yep.
And you move down the path.
Yeah.
And look, it's the way it's done. You can't sit around and say, "Geez, let me think of what I could do here." Novel.
Yeah.
Looking at something without having other information come to you.
No, you're absolutely
and
right.
Put things in a framework. We'll put it that way.
Well, I mean, I think back to one of the very first times I walked in here. One of the many times that I've walked in here to interrupt your day with any number of questions. And I remember maybe sitting here, I'm only meant to be in here for maybe like 2 minutes, and I think I was in here for 2 hours. And we were just asking a bunch of questions against each other about Alzheimer's and chlamydia in general, because I had a knowledge of microbes and microbiology and genetics and how these things interact with each other, and you had the knowledge of the brain, neuropathology, neuroepithelium.
Yeah.
And we were just discussing the topic, and that has led to probably like 4 or 5 different projects that have been forgotten at this point.
Oh, we'll remember.
Yeah, we'll remember at the worst time possible.
That's a good point.
We like to make the joke around here, put it in the notebook, and that's the notebook that no one ever gets to see. It's the notebook of all the ideas, and I think that's the notebook of it's so many different things that just aren't worth looking at.
Although it would be nice to explore these things as soon as you
Yes.
start talking about them, but unfortunately, very few labs have the capability of doing
We have to be targeted a lot of
Yeah.
times in what we're doing. It's not that any one question is more important than another. It's more, well, I know how I can look at this.
Yes.
And this might lead me to the next thing, which might lead me to the next thing. And that's the more, I want to say, advantageous way of kind of going at it. You're not discounting any one idea, but you're saying, I can do this one.
Yes, and over time you get to these different ideas. You start doing experiments addressing the different ideas,
Yep.
knowing that they all have a sort of place and time.
Yeah.
Some of it I feel like it's much too slow. I want to do it now versus wait
Yeah.
a month from now. That kind of thing. But look, there's everything comes at you at one time, and you have to sort of pattern it out. But
One question that got me recently, you remember Dr. Vignesh? He was on our show recently, back after he had his dissertation.
Oh, okay.
Shortly thereafter, I wanted to give him some time to kind of rest and recuperate, because performing PhD research is very difficult, as you know. And we kind of got to talk about that when he was on, but one of the things that he asked me towards the end of his time with me is, he said, if you had unlimited funds, would you do research on your own? Would you just go somewhere and do it? And I said, well, I looked at it from very much the business standpoint, and I said, knowing how many certifications and safety procedures I would have to put in place amongst the building itself, I probably would. Only because it's that idea, like you were talking about. I have all these ideas, and I kind of want to do them right now.
right.
But we almost can't.
That's true. It goes to the collaboration issue
Yes.
then, too, in consortium.
Yep.
Because that's the way to get everything done.
Yes.
Because another laboratory could maybe do it now, right? So as you talk about these issues, another laboratory may be doing, one experiment.
Yep.
Second laboratory, a second experiment, a third experiment. As you go to the different
Yeah.
laboratories. Okay. But all together, you're now addressing the problem, right? So,
When we write up grants and prospectus and proposals, we talk about specific aims. And a lot of times those specific aims are in and of themselves projects.
yes.
And they have their own hypotheses. And we expect specific outcomes that feed into the next aim and the next aim. But when we have these kinds of consortiums, one lab can be working on a single aim,
Yes.
while another lab is working on others, and so on and so forth. So it really does change the paradigm almost of how we're doing the science.
Yeah.
In a way.
It does, and it's much more efficient, I think, if you approach it that way as well. So
Well, you can dive deeper a little
Absolutely.
bit, because you don't have to worry about the idea of having to, okay, we've got to move it along because, we've got these other aims that we have to get done in time.
Right,
And you
right.
can almost allocate more funds to it that way.
You can designate it for the different projects now. And I agree. I think it's a better way of doing science. And it's also, it takes the ego out. I
Yeah.
think a lot of the times, but you do need to have different types of leaders within that to lead keep it organized so that it
Yeah.
all is maintains a fluidity to it. Realizing that, okay, you can share the accomplishment.
Yes.
That it's interesting because it's sort of like some people want to be a first author, some people want to be a last author, and to me it's about your contributions to the work is what's important. Okay, at times you can be a first author, at times you'll be a last author, middle authors, whatever.
Yep.
It's the contributions though that make the difference.
Yes.
And
Even
you have to think of it
scientists-
that way.
Even scientists argue about who gets top billing.
It can happen. Often it's discussed up front, so that if somebody is leading a project, then
Yes.
they would get top billing as the project evolves to a manuscript.
Right.
Okay. If somebody is a contributor but not leading, they may be a middle author. That type of thing. And usually that's acceptable for most people.
Yeah, that seems to be one of the aspects that I think has recently changed in science, and I kind of have to thank our Chinese scientific brethren, because they have hundreds of names on some of their articles. On this page here, for this consensus paper, there's a lot here of just names, but sometimes the articles that come out of Southeast Asia, they've got like a good 30 to 40 people just from one lab, and I think that has kind of opened the Western culture to giving more acknowledgement to the work that the technicians are doing, the work that the students are providing, the work that maybe someone who doesn't have that major degree now is getting that recognition for their contribution.
Yes, I would agree, and I think they should be recognized. It's important, for making contributions, and it does come down to that. This business can be one where obviously everybody has a level of ego. However, it's a humbling a thing to do. It's humbling because we're trying to make a difference for all of us.
Yeah.
Okay, and knowing full well that if we succeed, and when we succeed, that will be made. But in the process, there are millions of people suffering.
Yeah.
And if you keep that in perspective, what we go through here with deciding on an authorship of a paper is it's quite minimal, and in many ways unimportant in the overall effort.
Yeah, no, I completely agree with you, and that's another reason why I'm not trying to just be overly favoritist, but there's a way that this was written in terms of the authors and collaborators that I really enjoyed, and that was, it's written in here, the Intracell Research Group Consortium collaborators, and that allows for the group as a whole to kind of get the credit.
Yes.
Not just individual people like, okay, this person contributed this much and this person contributed this much.
That's correct.
But it's more, this is the group, and this is the group that did it, and here's their names at the very end.
Mm-hmm. Absolutely.
And I think that is very important. And I also have to highlight, one of the group that I work with as well. She's at my level, if you will, and that's Christine Hammond.
Correct.
She has done a lion's share of the technical work from your lab.
Yes, she has, and offered not only the technical expertise, but also the insight into the work and the perspectives. And often we joke about it because sometimes we say, well, we're sharing a brain because
No, you're
the thought
absolutely
processes are very similar. And and there are times where years ago I was much more in the lab than I am now.
Yep.
And it comes down to you evolve in different ways as you progress in this arena. So you end up on the same page always as to what's being done, how's it being done, evaluation of the data, that type of thing, and analysis, and you work together. I mean, it's a collaboration.
It really is. It's a working partnership.
Right. And with you as well, Joe.
Thank you.
It's a working partnership. It comes down to that. You have to have that.
I tend to have my head up in the clouds a lot because, as my wife will tell you, it never turns off. It just keeps going, and sometimes, and I'll admit it, sometimes that makes it so that you don't focus on the task at hand, if you will.
It can happen.
But another thing that's kind of good about that, and it's one of the reasons I was talking with someone about this earlier today, it's one of the reasons why I enjoy being at the bench, because I can sit there and think about it. I don't have to worry about the politics that's associated with being a professor or in searching out grant money. I don't have to worry about that, and that's kind of the benefit of my position, but there's also detriments there.
Well, what's interesting too is I went into science because I liked doing the experiments, and I still like doing experiments. And it comes down to that I do fewer this day and age than ever before.
Yep.
But that's the fun of actually doing science.
Yeah.
That's where you get to use what you're doing. You get to see it. You get to touch it and feel it. It makes a difference.
Yeah. I made a joke earlier today with Dr. Adams that he's unable to be in the lab because he's bogged down by a lot of administrative work. Right. Administrative work, teaching mentoring students, but also he's handling a lot of the study sections. He's handling a lot of grant review. He's handling a lot of the writing process. And I said to him, I made a joke, and then I said, and here comes me interrupting you going, I got a really bad image today. And the joke was that he would just take his hands and throw everything off the table and go, let me see. Because that's kind of what we as scientists enjoy. Yeah. It's that I don't care if it's bad. I want to take a look at it.
Right.
I want to see it because the more eyes that get on that image, like for you, the more eyes that get on that image, you might see that this organism is now there when it originally wasn't thought to ever be there. Right.
Exactly. It's good perspective.
And then that goes back to one of the things that you and I talked about at the very beginning. I truly believe that anybody can do scientific research, given that they kind of put their mind to it, if you will.
Yeah, and with the right training, the right mentoring and advising, and interest. Yeah, you have to be interested. You have to be curious. You have to be willing to think about a problem.
Yes.
And try to be creative about it, of really trying to figure out how do you answer the problem? Where's the solution, right? And knowing full well that it may not be an easy solution. It may be a very difficult problem.
And it can be even equally confounding because you might have more questions than you started with, more unknowns than you started with. And now it's like, well, I've got this result, but what does it mean?
That's right.
That I love whenever students come to me with that kind of reaction. It's like, I've got all this stuff. Now what do I do?
Right. Good point.
It's almost more interesting for me to see them react that way than it is for me to see them actually get the result that they're looking for.
Yeah, it's true. I look for the enthusiasm. Are you enthusiastic about doing this type? Looking at it that way versus, oh no, it's it's too boring, or I've got to wait an hour for something to incubate, and But to me, that's part of the excitement too, because you're waiting to see an outcome. Mm-hmm. You're waiting to see something of a result from an experiment.
Yeah.
And then of course, as you're saying, deciding, well, okay, now that I have this, what does it mean? Is it what I'd expect, or is it something different? How do I explain it? That type of thing.
There's a scene in the claymation movie Nightmare Before Christmas where he's doing all of these experiments on Christmas items, and he goes, "Interesting reaction, but what does it mean?" And I tend to play that a lot for students because it's like this is what you're going to be doing. You're going to do something, and because at the level that a lot of students tend to come into the labs for, they don't have the context that leads them instantly to that conclusion.
Yeah.
Based on what they're seeing. Case in point, we have two students that are working on a project with you and I,
right.
And they're staining for tau, and it didn't come up as intense. And they look at me and they're like, "Well, did we do something wrong?" I said, "Well, what does it mean?" And they kind of look at me like I'm nuts, like, "We want to know from you." And it's like, "That's great, but I want to know what you're thinking."
Right, exactly. Well, that's a good way to push it back to them. Say, "Okay, well, did you do something wrong?"
It doesn't matter what I think, it matters what you think.
Right.
And they look at me and they go, "Well, we followed the protocol." And it's like, "Okay, but did you make a mistake?"
Right. That's funny. It's funny.
No, and that I think that's the beauty of our chosen field in a way.
Yes.
Is that we can do the same exact technique over and over again, and yet achieve a different result every single time.
Well, hopefully there's a level of consistency.
I would hope, but sometimes it's not always that way.
Right. Well, that's true.
And sometimes entire experimental designs and protocols have actually been designed on the fact that they don't operate the same exact way in every single time, because that change in condition is the result in of itself.
Right, and that's where the development of protocols comes in, though, too. Mm-hmm. Is that's a trial and error approach as well. Sometimes you go, "Oh, well, let me try it this way," or "Let me incubate for 20 minutes versus 30 minutes," or "Let's use it at this temperature versus that temperature."
Right.
You can change all of those types of parameters, and at times get a better depiction versus a lesser depiction.
Yeah. And I think it's another reason why these kinds of consortiums kind of bring it all full circle are such a good thing, because you have that ability to say, "I've tried it this way," and it reminds what it reminds me of? It reminds me of the first lab meeting I was ever in, and I actually got to contribute and had to give a presentation. And even though it was a competitive lab and we were all jockeying for our PI's attention, at the very end of it, when it was all said and done and we all got to relax, the same person who tore me to shreds came up to me and said, "I think you could fix it by doing this, this, and this." And I simultaneously saw the good and the bad of science in that one meeting. We, like you said, it's the ego. We kind of jump over each other for the ego, but at the same time, we're also there to kind of help each other along, to answer the questions a lot of times. Even the most selfish of us are like, no, you didn't do it right. You got to do it like this.
Yeah, it can happen.
It can happen.
That's right. Well, that's where you have to be able to accept criticism in a positive way.
Yeah.
Okay, and that's because we're only human. At times we'll make mistakes, or there'll be something that we're missing, and if somebody else sees that and tells you that, you need to accept. Oh, okay, I understand now.
Right.
I can do it this way. That's just an acceptance of recognition that okay, there are going to be times where you may skip something because you're thinking about something else when you're doing your protocol, right?
Yeah.
And you didn't realize it.
So, happens.
It happens. It happens to everybody. Right.
No, it's true. And again, I congratulate you
Thank
on
you.
this consensus article. I congratulate not only yourself, but Nikki Schultek, Richard Lath, and the rest of the members of your consortium.
Yeah. Thank you.
I can't praise you enough for this level of work. And it's a very long review article, even by review article standards. But it's necessary. And I thank you for that effort.
And it's in some ways it's just the start.
Yes.
It's outlining what we need to do now, and now we've already gone into some planning stages now of how to move forward. Mm-hmm. And many of us are talking now of, okay, let's now keep the momentum going. Let's move forward. What's the best way of doing that? Mm-hmm. Yeah. So we have a meeting actually coming up later this week, I believe.
Oh, lovely.
So to talk about that.
Great.
Yeah.
It is currently 6:07, and as I always say to our audience here, this is at the end of your workday.
Yeah.
And I greatly appreciate you taking this time out of your own personal time to join us on this live stream. It's always a pleasure having you here, Dr. Balin.
Well, thank you.
And
it's a pleasure being here too. I enjoy it as well.
Oh, thank you so much. I greatly, appreciate you being willing to come on and talk about this consensus paper. I felt that it was very necessary to talk about. Yes. So, what's next for you in the research, really?
Well, we're for us here at PCOM, as you know, we've talked about a little bit potentially going into a different animal model.
Yep.
And looking at some other experiments that we can start to do. Also, the extraction protocols that you'll see in the consensus protocol is that I'm going to lead that effort here at PCOM in looking at different ways of finding the best way to identify the molecular signatures of infectious agents in different tissues and fluids of an individual. And we're going to go through different types of protocols to find those comparisons and make those comparisons and use that information, dispense it to our other collaborators and see if they'll use the same ones and find similar results.
Yeah.
So that's, and then others will do sequencing from their labs. We have lined up some interactions with some businesses, biotechs, and diagnostic companies to actually do some collaborative work. So we are already in the process here of moving forward that way.
Wonderful.
Yeah.
I think that's great, and I can't wait to read the next one from you and your group, because I think you're right. I think the momentum's there.
Yeah, we believe so too.
I think it's great.
So thank you for
my pleasure, Dr.
for inviting me to speak today too and talk with
you. You and I have said multiple times we could probably go all day, all night, and probably for the rest of the year without getting bored or tired. So do you have any lingering questions for me, or anything? Tell us what is. You have the floor, Dr. Balin.
Well, I appreciate you taking the time to speak with me about these issues, because they are so important for everyone to be aware of.
Yeah.
Where is this particular paradigm going? Why is it being developed this way? How is it going to be important for ultimately understanding what's really happening in a lot of these different diseases? And to me, that's extremely important. We need to know this. The science community needs to know it. The lay community needs to know this. We need to know that we're actually making inroads into something of great importance this way, that I truly believe is going to make the ultimate difference in combating these types of diseases.
Yeah.
And we're, obviously this is focused on Alzheimer's disease, but it is so applicable to so many others.
Yeah. Wonderful.
So thank you.
My pleasure. So let me take us home, and we will head on out for dinner or whatever we feel necessary. I want to thank everybody for joining us tonight. We definitely had fun. We always seem to. If you enjoyed the stream, please give us a follow. You can find this and other Science Talk episodes on YouTube. And the links are going to be in the description below, including the link for this article. Thank you, Dr. Balin, as always. And so long, everybody. Thanks for all the fish.
Thank you.
Good night now.